Okay, so last speaker of the session and of the entire conference is our own Virginia Ruetten. So.
Thank you to all of the organizers for the very engaging conference. I think a common theme that has emerged is that we have a joint interest in understanding how networks of genes or metabolic networks or networks of cells coordinate together to give rise to function and life. Classically, one might take the phenomena of interest. I'm not sure if this pointer works. It doesn't. I think I'm skipping the next slides. Thank you so much. Classically, one might take phenomena of interest, I think the pointer is still not working, be it cell motility or at the scale of the, I think not, sorry. We're frozen in time, but we're going to be trying to look at dynamics. Looking at cellular motility or at the scale of the organism, how an organism walks. Biased by the technology and the tools that we have access to, we might hone in onto a specific set of cells that correlate with the phenomena of interest. These cells correlate with walking, and through a set of perturbation experiments, we will find that if we ablate them, walking no longer happens, they're necessary, and if we activate them, something like walking happens, they're sufficient. And then we'll draw an arrow. Unknown to us, these are actually neurons in the spinal cord, and yes, they're an interesting and vital relay station, but because we weren't recording in the right place, we missed where a lot of the interesting computation and control was happening. Whilst not everything is important to measure in a system, identifying the critical nodes is non-trivial and needs broad measurement initially. And so the question becomes what to measure. I think the panel discussion yesterday was very interesting and highlighted really two things. One, the importance of dynamics, of density sampling through time, which microscopy lends itself to. And the second one, which was highlighted by Manu and also very well motivated by Eric Betzik, is the importance of context. That we can only really understand cells in their native context, in the context in which they function, ideally in tissues and ideally in the entire organism. While this is very challenging in most model organisms, the larval zebrafish offers us an opportunity for its transparency. And this has become a popular model organism in both developmental biology and neuroscience for its optical access. And neuroscience have really led the field in terms of imaging activity, which I would argue is maybe the closest thing we can measure to function. And through the use of genetically encoded calcium sensors, of which many have been optimized at Janelia, allowed to record the moment-to-moment fluctuations in calcium epoxy of firing in neurons. And this was really pushed to the limit by Misha Ahrens and Philip Keller at Janelia in 2013, where they developed the technology to be able to record whole brain functional activity, revealing numerous distributed computations across the brain. Of course, the transparency of the organism does not stop at the brain and extends to the rest of the organs, which as a vertebrate contains a lot of the same organs as you and me, be it a liver, kidney, pancreas, gut, and so forth. And so the question became, could we not extend some of the tools that neuroscience has brought to us to the rest of the body? With this in mind, I worked with the molecular genomics tool here at Janelia to create a transgenic line that expresses GCAMP, this genetically encoded calcium center, through all cells of the body. While calcium is a universal intracellular messenger, important in all cells, it still wasn't clear whether this would be useful. GCAMP has been optimized for neurons, and it wasn't clear if the dynamic range of these sensors would capture any interesting dynamics in the rest of the body. Thankfully, the very first experiment put a lot of these worries at rest, and I could see, under spontaneous conditions and no stimuli, a vast array, a plethora of activity across all tissues of the body. Here is just one plane, but I acquired these volumes volumetrically. After a few days of exhilarating experiments where you image everything that you can think of under all sorts of assays, you sit down and finally want to analyze your data. And here all of the problems begin. Unlike the brain, which is embedded in a skull, which constrains its motions, motion in the body is a lot less constrained. The gut twists and turns, sphincters contract, swim bladders deflate, making tracking any individual cell very challenging. The first thing in solving this was realizing that I really needed an additional channel, a reference channel, which simply labels the membrane, allowing me to visualize motion independent of any calcium changes. Then I teamed up with Wei Zhang, an engineer, and together we optimized an algorithm, classic machine learning, not really AI, based on estimating a flow field via an iterative algorithm that allows us to estimate a motion vector field, allowing us to register the data. Once the data is registered, I can then use variants of non-negative matrix factorization to do cellular scale segmentation, resulting in a data matrix of cells by time with functional activity across the body. Then the next problem arose. A lot of the value of this matrix comes if you're able to assign individual organ identity or cell type to each individual of the rows. So how can we do this? Taking a second look at our data, this time a little bit more closely, you can see that rather than being a sea of green, or in this case magenta, you can see that there's a lot of structure. And that with a little bit of practice, by eye you can start teasing apart what is the muscle, the spinal cord, the notochord cord that we heard about yesterday, and so forth. And that you can tell apart a surprisingly large amount of tissues or cell types. Of course, one could think about automating this with a pixel-level classifier or some kind of model, but this would have required a large amount of data, for which I neither had the time nor the resources to create. So instead, I wondered whether a more unsupervised approach could get me part of the way. And again, going back to this data matrix, we see that there's a lot of structure. And so I derived a variant of a graph-cutting algorithm that uses coherence as a similarity metric to essentially cluster activity time series that have similar temporal profiles, resulting in a set of about 400 clusters with varying dynamics. What this allows us to do is to then go back and look at the spatial footprint of these clusters, which thankfully for me mapped most of the time to individual organs, allowing me to then go back and annotate this data relatively efficiently. And here we can see that, for instance, we see oscillations in the enteric nervous system, bistable dynamics in the sympathetic nervous system, some ultra-slow fluctuations in the meninges, and so forth. But more interestingly, not only did the clusters map to individual organs, but more often than not, to individual compartments within organs. This is, for instance, the kidney nephron, which you can see contiguous clusters, even though the algorithm has no information as to the spatial provenance of the cells. And indeed, we see that the kidney has various temporal profiles, just spiraling, and then which propagates into a traveling wave, which indeed couple together. And further experiments show that, for instance, if you deliver drugs, you can see that this accelerates the frequency of these. However, one may ask, what is, for instance, the molecular identity of these different compartments? Or in other cases, for instance, I could identify clusters of cells that responded, for instance, to hypoxia, low oxygen levels, but I wasn't able to identify their cellular identity. While there are many high-quality atlases for the brain of the zebrafish, there is a paucity of literature on the rest of the body relative to the brain. And so I really needed to be able to have a high-resolution contextualized anatomical and molecular data to be able to interpret these functional data sets. Whilst there are many clearing approaches, they were never really quite satisfactory. And being at Janelia, I had the privilege to work with Paul Tilburg, a co-developer of expansion microscopy, to be able to develop a robust whole-body expansion microscopy protocol. Expansion microscopy is a form of super-resolution microscopy which works by physically expanding the sample, giving us a boost in, not only a boost in resolution, but also excellent clearing. As a result, we can have unhampered access to tissue all across the body of the animal, which before was difficult. Moreover, using total protein stains, which are commonly used in the field, these gives us rich contextual information about all sorts of aspects of anatomy. For instance, here we can see putative primary cilia, or microvilli, or the basement membranes of various tissues in the body, allowing us to then go back to the functional imaging and better interpret these. Moreover, the protocol is compatible with antibody, retains antibody signal, and critically enables us to also do fluorescence in situ hybridization, allowing us to really get the ground truth molecular identity of a lot of these cells. Together, this provides a whole organism live imaging system for recording tissue and intracellular activity that we abbreviate to holistic. So what can we do with this technology? In the next part of the talk, I'll briefly touch upon 3 broad aspects with just a few examples illustrating of where we're hoping to go with this. The first one is relatively simple. It's simply characterizing cellular responses to stimuli and drugs, but hopefully being able to do it at a scale that wasn't previously done. The next level of complexity would be looking at inter-organ interactions, and then really the holy grail is being able to really dive into physiology and look at circuits and control mechanisms. So for the first, this might sound relatively non-creative, but it's worth remembering that a lot of these cell types have never been functionally imaged before, and that just understanding the impulse response of a system or the sensitivity of cells is sort of a fundamental building block. And for instance, if we're maybe interested in what cells are responsive to cold, we can simply, as an example, deliver cold water to the fish, rank the cells in terms of responsiveness of various cells, and identify as one of the top hits this cluster that we can see here. Using some of the tools that previously mentioned, we can probe directly for the cellular identity of these cells and discover that these are actually chondrocytes of cartilage that are highly sensitive to cold, a property never before reported, and when you think of the number of people that complain of joint pain in winter, might be clinically relevant. This is just one of many examples. You can test drugs and discover that ketamine, beyond affecting neurons, very much affects the meninges, for instance, that your nephron is highly sensitive to increased temperature, and so forth. There's clearly more that what one could do in a single PhD, but I think given the number of, for instance, clinical trials that fail because of off-target effects, there would really be a worthwhile in simply atlasing and characterizing this, and that this itself would be a significant contribution. But next, where we really want to go is starting to look at inter-organ interactions. One can go back to our initial example of asking what happens when we move, and ask, when we move, we not only act on the world, but of course we're also affecting our internal biology. So what happens when skeletal smooth muscle contracts? We can fit a relatively simple kernel regression model and identify various features. For instance, that the notochord cells of Alex are highly responsive when the animal moves. But my attention was particularly captured by these hindbrain neurons, whose variance was very much explained by the specific sphincter of the gut. After a while, you want to go beyond correlation, and so then I included, to be able to add causal perturbations to the system, I could include optogenetics by expressing channel rhodopsin in motor vagal neurons, and then precisely activating cells that were in the same region to then simply image what happens in the body and identifying that indeed these neurons induce sphincter constriction, and then I could go on to tile the entire hindbrain and uncover a topological mapping between the hindbrain and the gut, and so forth. Briefly, because I realize that I'm running out of time, we can then finally start looking at more physiological circuits and ask, for instance, what happens when you start restraining the amount of resources that the animal has. For instance, when the animal has access to lower oxygen, a vital resource that all cells need. As before, we actually saw that there was this cluster that became more active. This turns out to be the sympathetic nervous system. And within the same experiment, one could see that a lot of the activity in the gut and the viscera became very, very quiet, but there was one cell type that became a lot more active, which was actually the cells surrounding the artery feeding blood supply to the gut. And that this, in subsequent experiments, very much contracted. But then one might ask, what does contraction of a vessel amount to? Is it actually changing the blood flow to any significant amount? So then I could image blood flow across the body. And these are 2 videos during either normoxia and hypoxia. And hopefully you don't see a lot of difference until I highlight where the vascular vessels are. And hopefully you can see that there's drastic decrease. We can again go beyond pure correlation and start manipulating the system in precise ways, such that, for instance, you might want to ask, is this a cell autonomous process or is this driven by the brain? And so for this, we can express inhibitory opsins in neurons. And at specific times, I can either do a control stimulus, nothing much happens, sorry, this is blood flow, it's decreased because there's hypoxia. And then once I inhibit the brain, blood flow is restored within seconds, and I can repeat this, really highlighting that this is an active diversion of blood flow in a neuronal-mediated process, and moreover, sort of highlighting that we've been able to, in very few experiments, uncover a circuit, and that moreover is correlated with reduced gut activity. And one can ask now, is this sort of a new control mechanism? Because we very much think that the brain controls the body either via nerves or via hormones, but another way could be simply diverting blood flow and reducing the amount of fuel that you give to the system. And so we're now, doing further experiments to prove this. And of course, the developing level organism is still developing, and we're keen on working with the SATU lab to be able to apply some of this technology to adult vertebrates. The Danuela cerebrum is a new model organism that a lot of people are excited about that would potentially give us the opportunity to do these sorts of recordings in a fully developed animal. With that, I'll take Manu's title, I hope you won't mind, with a slight alteration, and ask, can we do cellular biology not only at the scale of the cell, but maybe even at the scale of the body? And that now, with these data sets, I'm beginning to dip my toes into some of the more sophisticated models, of which I was very inspired to see so many people do work. And so it's very exciting to be able to go into this direction. And before closing, I'd just like to take a moment to reflect that neuroscience has made tremendous progress in really understanding how neurons collectively are able to learn, store memories, drive adaptive behaviors, but that it's taken decades to come there because the tools and the methodologies, the compute power, were all developing at the same time. Whereas I think we're in a remarkable position where all of these are converging, perhaps exploding at this point in time, and that we have the potential to potentially leapfrog what will otherwise be decades of fragmented work if we commit to collecting data at the scale and with the ambition of Eric Betzek, but for whole body functional data, and also having concerted effort of adapting models to such routine to be able to have, for them to be able to predict, for them to be able to query, that we can have in silico experiments to really drive and accelerate this discovery cycle. With that, Janelia really is a magical place in which you have the opportunity to work with a huge range of experts across multiple fields, and really allows you to go beyond what you could have ever done as an individual person. We are just one node in a network. And with that, I'd like to say a big thank you, first and foremost, to my advisor, Misha Ahrens, here, and also a lot of the other teams, the help with the expansion microscopy, the support and help for the transgenic line, all of the advice that I had on the optics, the help with the registration, the analysis, all the incredible sensors and dyes that we have access to, and a lot of other people that you can brainstorm with and help. And with that, thank you very much.